Okay, I guess we'll get started now then. Thanks everyone for signing up and signing in. It's really a privilege to welcome Dr. Reluca Gordon for today's QBI seminar. Dr. Gordon did her PhD in the Hardeman Lab in computer science at Duke, followed by a brief postdoc in Martha Bulick's lab, and working on really developing and applying new methodological tools to study protein-DNA interactions. And this is a theme that her lab is really, in a really exemplary fashion, kind of continued to follow as a first an assistant professor at Duke and now tenured professor at Duke. A bit of a personal note, I've always I've been a big fan of her work for a while now. I think first stumbled upon her work looking at using protein binding arrays to really dissect the context specificities of various paralogs of different transcription factors, and most recently was really excited by her most recent paper published in Nature last year that I think she'll talk about. So so with that, the floor is yours, Raluca. Thanks again for coming.
Thank you very much, Vijay, for the introduction and for inviting me. Let me share my screen here. Okay. Can everyone see this?
Yes.
Okay, thank you. Okay, so as Vijay said, my lab is interested in studying protein-DNA interactions, and we're specifically focusing on transcription factors. At Duke, my appointment is in biostatistics and bioinformatics and computer science, but I'm not really a biostatistician. And although my training is originally in computer science, I don't think I'm a computer scientist either. Our physical home for our lab is in the Center for Genomic and Computational Biology, which is actually located in a building located very close to the Duke Chapel in the beautiful campus. I'm also part of a number of centers, and I have a secondary appointment in molecular genetics and microbiology. And this pretty much reflects the interdisciplinary nature of our work, and the fact that we're focused on answering questions about transcription factor binding. We're not so much focused on a particular discipline or technology or methodology of any kind. So I don't have to convince this audience that genomic binding of transcription factors is very complex. We know sometimes proteins bind directly to DNA to regulate their target genes. Other times they bind cooperatively to DNA, or they bind to individual sites and cooperatively regulate a gene. There are also indirect interactions and competitive interactions, especially between paralogs that bind to similar sites. And in our lab, we are developing quantitative techniques, to both computational and experimental, to study this genomic recruitment of transcription factors, mostly in human, and to understand how this is affected by genetic mutations and more recently by DNA lesions. There are kind of three major research themes in our lab. One is to decipher the codes that govern the genomic recruitment of transcription factors. And at the moment, two students in the lab, Yuning Zhang and Vincentis Martin, are working on cooperative and competitive binding of transcription factors to DNA. We're also trying to understand how genetic mutations and variants can affect transcription factor binding and subsequently gene expression. And we are developing a tool that we call QBIC to make quantitative predictions on the effects of genetic variants on transcription factor binding, trying to take advantage of the statistical expertise in our lab and making sure that we incorporate implicitly the quality of the data in our predictions of the effects of mutations. And a third direction, and this is what I'm going to focus on today, is the interplay between transcription factors and DNA damage and repair. And this is a direction that we started a few years ago. Many projects in this area have been started and driven by Ariel Affect, a very talented postdoc in our lab, who will start his own lab at Weizmann Institute this fall. And we have a number of graduate students now working on various aspects of how transcription factors interact with DNA damage and repair, since they both act on the genome at the same time. As a general strategy, in our lab, we take this very complex environment in the nucleus, and we try to reduce this complexity by doing experiments on chips or glass slides, or sometimes we call them microarrays. We try to stay away from the word microarray just because some people will think these are obsolete. And indeed, for gene expression studies or things like chromatin immunoprecipitation, microarrays are not used that much anymore. In our lab, we have a very different strategy for using these microarrays. I was describing some of our work last week to Bruce Tillman. I had the honor to meet him. He gave a talk here in biochemistry, and he summarized it as us doing biology on a chip. I would refine that a bit to say that we're doing quantitative biology on a chip. So basically what we do is we take the genomic regions of interest and we synthesize them on a glass slide as single-stranded DNA. And this can be done commercially, which is what we do now, for example, by Agilent. And these sequences can be anything, so they really depend, the DNA library depends on the questions we're asking. Once we have the single-stranded chip, we double-strand it, and we have different protocols and strategies for doing that, depending on the question we're asking. And once we have the double-stranded DNA, we can do protein binding experiments. We can look at direct binding of one transcription factor to DNA, cooperative, competitive binding, and we can also quantitatively measure the effect of mutations and lesions. With this kind of technology, we can get quantitative measurements for transcription factor binding to tens of thousands of sites at the same time. So after we incubate the proteins of interest with our DNA library attached to the glass slide, we can either use a fluorescent tag on the protein, or we can use fluorescent antibodies that recognize the protein or a tag on the protein and measure quantitatively how much protein we have at each DNA spot. I have to say that these experiments that we do on chips were inspired by the PBM technology that I learned as a postdoc in Martha's Bullock lab, and I'm very grateful for everything she taught me and also for her mentorship. One reason why we really like these on-chip experiments, and for the most part, we haven't switched to sequencing for a lot of our data, is because the on-chip data is very reproducible and quantitative over a wide affinity range. So here on the left, I'm showing a replicate plot, and this is from our recent cell systems paper where we looked at the specificity of transcription factor paralogs. And the data is showing replicate binding in vitro for MAD protein. This is a human transcription factor. And every black point here is one genomic region centered on a MAD binding site, while the gray regions are just negative controls. And we can see that the in vitro binding level, as measured on these chips, is very, very reproducible. Now, of course, this is one of the best replicates we have with an R-squared of 0.99. But in general, for technical and even biological replicates, we get R-squared values of 0.9 to 0.99. We're also happy to see that our measurements, which are basically fluorescence intensities, agree very well with independently measured equilibrium dissociation constant. And this is another, on the right here, there's another figure from our cell systems paper where we compared our fluorescence intensity measured for transcription factor max against MITOMI data that was available from Stephen Quake's lab. And again, we see a great agreement, an R-squared of 0.91 over a range of KDs between 1 nanomolar and 1 micromolar. More recently, in the paper I'm going to focus on today, we also did a number of comparisons looking at how our fluorescence intensity, which reflects the level of binding of a protein, correlates with independently measured affinity dissociation constants. And for a wide variety of proteins, we saw excellent agreement between our measurements and KDs measured using fluorescence anisotropy, surface plasmon resonance, EMSAs, Mitomi, pretty much any external data that we can get our hands on, we can use, and we see very good agreement with our binding measurements. Now, what I'm going to focus on today is a paper that we published last year in Nature on transcription factor binding to DNA mismatches. And just to clarify from the beginning, by mismatches, we mean non-complementary Watson-Crick base pairs. Like an example here would be TG, so any base pair other than AT and CG. And we are very grateful to the reviewers, including James and Kale, who also wrote a News and Views article about our paper. The reviewers were really helpful in improving this work. I would also like to mention that we submitted a proposal for a cover image that you can see here on the cost of distorting DNA. Now, unfortunately, Nature did not choose our cover. They chose this picture showing patterns in evolution. But we were happy to see that they highlighted our paper right after the evolution, coronavirus, and climate change. So if those are the papers that were highlighted before ours, we cannot be too upset about it. So many times when we talk to other scientists about our work, they asked us why did we even think about measuring transcription factor binding to DNA mismatches? Because we know transcription factors because of their role in gene regulation, and not so much as having anything to do with DNA lesions. And the answer is a bit convoluted. We actually started thinking about DNA damage more broadly. And we know that DNA damage occurs frequently in the genome. So when transcription factors bind to the genome is also exposed to a number of different damages. And repair is also happening on the genome at the same time that transcription factors bind and regulate their target genes. So it's not unreasonable to ask how transcription factors bind to damaged sites. We know that the damages will distort the DNA, and we know that transcription factors are sensitive to the DNA structure, so we can expect a significant effect here. Now, one could argue that the genome is large. We have 3 billion bases, and transcription factor binding sites are short, just about 10 to 20 bases. So maybe the probability that a damage actually hits a binding site of a transcription factor is very small. And we could think this, but actually if we look at mutation data, and specifically at somatic mutations observed in cancer studies, we see that there's an enrichment of mutations at transcription factor binding sites. And there was a beautiful paper from Rial Lopez-Bigas's lab in Barcelona. This was published in 2016 in Nature. And this was largely the inspiration behind our work. So what Nuria's lab found is that looking at somatic mutations in melanoma and also a few other cancer types and also skin cells, they saw an enrichment of mutations at transcription factor binding sites. So this is more mutations than expected by chance, and more mutations that we see in the flanking regions of transcription factor binding sites. And in addition, they saw that not only there is an enrichment of mutations at the binding site, there's also a depletion in repair. In this case, they were looking at UV damage and nucleotide excision repair. But they posed this hypothesis that transcription factors, when they bind to their binding sites, if those binding sites are damaged, then the damages will be less well recognized by repair enzymes. And in that way, the transcription factors will have a negative effect on DNA repair. So we might see less repair at transcription factor binding sites, and this might be the cause of the increased mutation rate. Now, this was a beautiful study and very inspiring, but it was based on correlations between different genomic data sets. And there are still a lot of questions that remain. Importantly, we started asking do transcription factors even bind to damaged DNA? If they don't, then there's no competition with DNA repair. If they do, then the question remains, what are the rules governing this binding? Is any lesion going to be bound? How are different lesions going to be bound with different affinities? And importantly, how exactly will transcription factor promote mutagenesis if they do bind to these lesions? Where in the repair process are they going to have an effect? So this paper was focusing on nucleotide excision repair, but we thought that if this is a more general mechanism, then we would expect a similar effect on mismatch repair and base excision repair and so on. Not long after this paper came out, we started a series of projects focused on the interplay between transcription factors and DNA damage and repair. And as I mentioned, Ariel is the one who drove many of these projects as we started. And this paper that we published last year is the first paper from our lab in this new research direction. Thinking about what lesions we want to study, DNA mismatches made a lot of sense to start with. Importantly, we know that if we look at cancer mutations, for example, it's been estimated that about 2/3 of cancer mutations actually come from unrepaired replication errors, which are mostly mismatches. So it did make a lot of sense to look at mismatches first. Looking carefully at the literature, we found basically one previously published paper on transcription factor binding to a mismatch site. And these were CC mismatches. And not only that, this paper was from Jack Griffith's lab at UNC. So especially given the tension between Duke and UNC, it was a must for us to go ahead and study transcription factor binding to mismatches and characterize this recognition. We know that in the cell, mismatches can occur due to nucleotide misincorporation by DNA polymerases. And oftentimes we think of this happening during replication, but it can also happen during repair of other damages. And we know, for example, that gap-filling polymerases are more error prone. So they may generate mismatches as they repair other damages. Mismatches can also occur from spontaneous chemical reactions, such as deamination of 5-methylcytosine, which results in TG. So there are several sources of mismatches in the cell. Looking at what mismatches do to the DNA, they definitely alter the structure of the DNA. For example, they can change the major-minor groove geometry, and they can also affect specific local parameters of the DNA. In addition, they change the placement of functional groups that are available for transcription factors to bind. And they also reduce the stability of the double helix. For example, CC mismatches are very easy to melt. Now, given that transcription factors care about the structure of the DNA, it wasn't clear to us how mismatches will affect transcription factor binding. There wasn't any data in the literature to help us build some kind of predictive model, because the structures of mismatches are very different from the structures of Watson-Crick base pairs. So because we really didn't have any data to work with, it made sense to develop an assay to measure transcription factor binding to mismatched DNA. And we developed this assay called SAMBA, Saturation Mismatch Binding Assay, where we can take transcription factor binding sites. They can be artificial sites, they can be sites from the genome, and we can test several sites in a single experiment. Once we select our sites, we can take one of the strands and keep it as wild type and order it in solution with a small amount of size 3 label. For the second strand, we generate computationally all the single strand variants and synthesize them on a chip. Then we can hybridize the oligos in solution on the chip to obtain double-stranded DNA. And in a way, we can think of this as a dance of the two strands, and this goes well with our assay name, which is Samba. Once we do this hybridization, we can check the efficiency to make sure that we have uniform hybridization across the different sequences that we're testing. And we see this by using a microarray scanner. The black dots here are negative controls where we did not expect any signal. After we confirm that the hybridization worked well, we do a protein binding experiment, pretty much like a PBM, where we incubate with a transcription factor and with an antibody that is labeled with a fluorophore. Then once we scan our chip, we get a picture like this, where bright spots represent DNA sequences where a lot of molecules of the transcription factor were bound. So each of these spots contains millions of copies of identical DNA sequences that we designed computationally. And if we have a lot of DNA molecules bound to that particular spot, then we get a bright signal. If not, we get signals of different intensities. The reproducibility of this kind of data, as with other chip assays, on-chip assays, is very high. In this case, for example, in one replicate experiment, we got an R-squared value of 0.98. So it's very, very reproducible. The first mismatch binding experiment we did was for a transcription factor called ETS1. And in here, we took a binding site of ETS1. We can take these from the genome together with their context. And computationally, we generated all the possible variants, in this case of the upper strand, while keeping the lower strand constant. So we have three variants for every position that we want to mutate. And this rectangle here is just showing the location of the binding site. Because we have tens of thousands of spots on these arrays, we can do multiple replicates of each sequence. And in general, we do between 8 and 20 replicates. So we get a distribution of binding levels for every individual sequence that we test. In here, I'm showing some of our data for ETS1. The y-axis show the log2 of the ETS1 signal. And this is just arbitrary units because it's fluorescence intensity. The black bar here, the blue bar represents the binding levels we obtained for replicates of the Watson-Crick site. And the box plots represent the data for the individual mismatches that we tested, focusing on the mismatches in the binding site itself. Because we have many replicates of each sequence that we test, we can ask for any mismatch of interest if the binding level is significantly different from the wild type. And we do this using a Mann-Whitney U-test, and we also correct for multiple hypothesis testing. A nicer way of visualizing this data is using these mismatch binding profiles or Samba profiles. And this is the profile for transcription factor ETS1. Here on the y-axis, again, we have the log2 of the mismatch signal divided by the wild type signal. So at 0, we have the wild type site that was Watson-Crick. And for this strand shown here, we make all possible single strand mutations. So all of these are the mismatches that we tested. Mismatches that are above the 0 line show increased binding, below the 0 line show decreased binding. And here we also color them based on their p-value. So only mismatches that are significantly different from the wild type are shown in color. As I mentioned here, we keep one strand constant, and this is the bottom strand, and we mismatch the upper strand. As expected, we found that in the flanking regions of the binding sites, mismatches have little influence on binding. We rarely see any significant effects from the mismatches, with some exceptions, and I'll come back to this G. This is actually a GA mismatch, and I'll talk about it a bit later. For the most part, we don't see much of an effect in the flanking regions. Also, as expected, the mismatches in the binding site do have strong effects. Now, our expectation was that all mismatches will likely destroy binding. We do see that a lot of mismatches do decrease binding. Also, they decrease binding to different extents, and we see a wide range of effects here. The big surprise for us was that we saw mismatches that enhanced binding. And for ETS1, particularly these 4 mismatches located directly in the binding site of ETS1 had a significant increase in binding. And just as an example, this T here actually was a G in the Watson-Crick site, and we mutated to a T and thus obtained a TC mismatch at this position in the binding site. It might be hard to figure out the magnitude of these effects because we're just showing here log-to-fold changes. We did independent measurements, and for ETS1, we collaborated with Gregory Poorn's lab at Georgia State, and his student Suella did KD measurements using EMSA, using fluorescence anisotropy. In this particular case, for the mismatch that had the largest effect on ETS binding, which is an AG mismatch shown here, the increase in KD was from 15 nanomolar to about 1.6 nanomolar. So about 1 order of magnitude increase due to this particular mismatch. So the effects are not negligible. It's not a small increase in binding. We did additional validations for other proteins. We also collaborated with Tali Haran's lab at Technion and Miles Bufol at University of Iowa to look at P53 and GR binding measurements. And again, we validated very nicely that the measurements of increased and decreased binding on our chips agree very well with independent KD measurements. Given this very good agreement between our signal intensities and the KDs, we can actually use this, the few KD measurements to calibrate our data. So all the tens of thousands of fluorescence intensity measurements we have, we can transform them into KDs. We did this for P53, ETS1, and GR using measurements to mismatches. But we can also do this using KD data from the literature. And in this case, we also generated libraries for Watson-Crick sequences, and we were able to calibrate CBF1, MAX, TBP, and EGR1 data. Initially, when we saw these results for ETS1, and we noticed that increase in binding that was also significant, we thought that maybe ETS1 is a special transcription factor. For some reason, it likes a few of the mismatches. So we did test a lot of other proteins. In the paper, we talk about 22 transcription factors that we tested. We have a few more that we've done in the lab. And pretty much for every single transcription factor we test, we find some mismatches that increase binding. The identity of the mismatches is different. The position, their exact position in the binding site is different. But we see this kind of increase for every single transcription factor that we test. And this included yeast transcription factors, Arabidopsis transcription factors, human, mouse. They all have some mismatches that increase their binding. In addition, looking more closely at the data we generated, we also found examples of mismatches creating binding sites, sometimes on a single strand. So in this case, this happened for the mismatch that we saw increasing binding in the flanking region of this ETS1 site. Now, initially, when we saw this, we got very excited seeing some of these mismatches in the flanks, increasing binding. We thought that there might be an interesting mechanism where there's some allosteric effect through DNA, so that whatever distortions this mismatch does will propagate and improve binding of ETS1 to its site. It didn't seem to be the case, though. If we look more closely at the sequence here, and we look at the particular G that was mismatched, this is a GA mismatch. But now looking at the strand where the G was introduced, this becomes a GGAA site, which is the reverse complement of TTCC, the preferred core binding site for ETS1. And also looking further at the structure of ETS1 bound to DNA, we know that contacts between ETS1 and the DNA do happen at these bases. So in this case, we can think of the mismatch as an intermediate step to the Watson-Crick mutation, and the Watson-Crick mutation is also bound very well by ETS1. So for this example, we don't think there's any allosteric effect, but more the creation of another site, although on only one strand. Now, it could be that in general, what these mismatches do is just act as intermediates to the full Watson-Crick mutation. But from our data, that does not seem to be the case. In fact, mismatches and mutations have very different effects on transcription factor binding. So I've already shown this mismatch binding profile for ETS1, where these 4 mismatches in the binding site increased binding. We can also generate a DNA library of Watson-Crick sequences to look at the effect of ETS1 mutations. And we see that if we focus on these 4 mismatches that increase binding, the corresponding mutations actually decrease binding. In the mutation data, we saw 2 mutations that increase binding, but here the corresponding mismatches decrease binding. So overall, if we try to look at a potential correlation between the effects of mismatches and the effect of mutations, there isn't a good correlation. And we definitely see a lot of outliers where the effects are opposite. Just to give 2 examples here, this G mismatch shown here is actually a GG, and the initial Watson-Crick base pair was CG. So although we see an increase in binding from the GG mismatch, the corresponding GC mutation decreases binding by about 2.5 kbt. And this is also the case for the other intermediate mismatch that we could generate, CC, which also decreases binding. As another example, at position 4 here, we have this T, A to T mutation. So if we go from AT to TA at position 4, the binding increases, but none of the intermediate mismatches, TT or AA, shows an increase in binding. They both decrease binding. So from examples like this and from all the massive data that we generated, it's clear that the mismatch effects cannot be explained simply by base changes. Mismatches are not one step towards the Watson-Crick mutation. So with all the SEMBA data we generated, it became very clear that transcription factors can bind mismatches with high affinity. We've seen a lot of examples where the mismatch doesn't really have an effect, and about 10% of the mismatches we tested in the binding site actually increased binding. So then we started wondering what mechanisms could lead to this unexpected increased binding to mismatches. And because just sequence analysis didn't lead anywhere, it was clear that it's not a simple sequence change. And it had to be DNA structure or DNA dynamics. We reached out to Hashim Al-Hashimi in Duke, in the Department of Biochemistry, and that's just across the street from us. And we worked very closely with 2 students in his lab, Atul and Honglu, who together with Ariel did a lot of structural analysis to help us understand how these mismatches actually increase transcription factor binding. The reason why this increased binding was a surprise to us is because we know that mismatches change the structure of the DNA, and we thought that that would have a negative effect on transcription factors. So if we look, for example, at a GC base pair, and we transform it into a GT mismatch, we create a shear in the DNA, and there's also a slight bend in the DNA. If we take the GC and mutate it to a TC, then the diameter of the DNA narrows because these are both pyrimidians. And given how much transcription factors recognize DNA shape, we really thought that these kinds of deformations would negatively affect TF binding. But a full analysis of all transcription factor DNA complexes in PDB revealed a lot of examples where in bound DNA, we see deformations that are very far from ideal B form. As we show in our paper, around 40% of the complexes we analyzed from PDB show at least one position where there's a deviation beyond what we see in B form DNA in its naked form. And here I'm just showing some examples that we also have in one of our supplementary papers where pretty much all the transcription factors that we used in our study has had some form of deviation from bDNA. So these deformations, surprisingly, are not that rare in transcription factor DNA complexes. And in addition, in some cases, the distortions introduced by protein binding can also be achieved in naked DNA using mismatches. So just to give you an example, a GT mismatch can mimic protein-bound sheared DNA. So here I'm just showing a regular Watson-Crick AT base pair. But in certain protein-bound structures, the AT is sheared. Looking at the structure of a GT, we realize that this actually aligns very well with a sheared AT base pair. So it's not a perfect alignment. The backbone is a bit different, but it's surprising how similar the GT mismatch in its naked form is to the bound sheared AT. And we found several examples of these. Of course, the picture is complicated because mismatches change a lot of local structural properties of the DNA. But this led us to the general mechanism that we describe in our paper. So basically, when we think about protein binding, the energy comes from two major terms: a penalty for distorting the DNA, and a gain in energy because of the interactions that are formed between the transcription factor and the DNA. And here we're not making any claims about the order of these events, but just that the DNA must be deformed because in its bound form, it is deformed, and we know that needs to happen. Distortions, of course, will have an energetic cost. When we look at mismatches, if a mismatch can pre-deform the DNA even before protein binding, then it effectively prepays some of the energetic cost of this deformation. In the end, this results in a gain in binding affinity. Analyzing all the data we have and all the mismatches for which we saw enhanced transcription factor binding, we found some form of structural mimicry in about 2/3 of cases where the mismatches increased binding. So we really think this is a general mechanism. And because our data is so quantitative and we can map it to KDs and to binding energies, we were able to conclude that this penalty for deforming the DNA is an important part of protein DNA recognition. I'll focus also on two examples that we also describe in detail in the paper to kind of illustrate this mechanism. A very nice example is that of P53. So looking at structures of P53 bound to DNA, we know that there are certain positions in the P53 binding site where the DNA is constricted. The bases in these regions are Hooksian base pairs, and the diameter is reduced by about 2 angstrom. At exactly those positions, when we looked at our SAMBA data, we saw an increase in binding due to TC and TT mismatches. So these are two positions where the DNA is constricted, and these specific mismatches increased binding. We didn't see much increased binding anywhere else, and TT and TC definitely did not increase binding at any other positions. Looking at the structures of TT and TC, we know that compared to the Watson-Crick AT, which has a diameter of about 10.6 angstrom, the TT and CT reduce that by about the same amount as what we see in Hookstin AT, which is what the P53 bound form is. So the same reduction in DNA diameter is achieved by these specific mismatches even before the protein binds to the DNA. So that easily explains why we would see this increased binding. We did a lot of other validation experiments here, and in collaboration with the Al-Hashimi lab, we also did NMR to show that these mismatches truly mimic the Hookstin AT. And those experiments are described in our paper. As a second example, I'll refer to TBP, the Tata box binding protein. TBP bends the DNA at the beginning and end of the binding site, and specifically at the 3' end of the site, there's a very unstacked base step where TBP introduces a phenylalanine in between these two base pairs. And the stacking is really affected at this particular position. When we did our Samba experiment, at this G at position 8 here, we saw increased binding due to mismatches, pretty much any mismatch we tested. And specifically, we saw a large increase in binding if we put a CC mismatch instead of the normal GC base pair. And in this case, the Watson-Crick site is about 15 nanomolar, while the CC mismatch is about 5 nanomolar. So looking at what CC mismatches do is they are very unstable. There are a few structures of CC mismatches. There's barely any hydrogen bonding between them. And definitely we see reduced stacking when we have CC mismatches. So again, it made sense that this reduced stacking, which we also find in protein bound, in TBP bound DNA, is the reason for the increase in binding affinity due to the CC mismatch. More broadly, we were actually able to correlate the transcription factor binding energetics, so the KD, calibrated from our data, to the stability of the free DNA. So we can use nearest neighbor energetics to predict the stability of the DNA when we introduce certain mismatches. So this is the regular binding site with a CG at position 8. And this is the CC, the mismatch that is most destabilizing, and it was also bound with the highest affinity. And we were nicely surprised to see this very good correlation between the stability of the duplex when we introduce different mismatches and the affinity of binding of TBP. And again, here we refer to the stability of the unbound DNA, so before it encounters the protein. We saw this kind of correlation at several positions in the TBP binding site. The largest correlation was at position 8 here, but also position 7, which is part of this base step where stacking is highly affected. We also saw some correlation at one of the positions at the 5' end of the binding site, where again, there's DNA bending and some unstacking of the base pairs. Furthermore, we validated here that in the bound form, the Watson-Crick binding site and the mismatch binding site are nearly identical. So we collaborated with Maria Schumacher also in the biochemistry department here at Duke. We are lucky to have wonderful collaborators just across the street. And we selected a couple of mismatches that were strongly bound by TBP, including AC and CC mismatches, and performed X-ray crystallography to determine the bound structures of TBP with AC and CC mismatches. And we found an excellent agreement between these structures, there's barely any difference. Definitely no difference, like no added hydrogen bonds or something that would explain the increase in binding affinity due to the mismatches. So this was really important for us to see, because it means that this difference in affinity of binding due to the mismatches is not because of the bound form, at least not in this case where we have a crystal structure of TPP bound to mismatches. The difference in affinity actually comes from the unbound form and this deformation that needs to happen, which is easier to do in the mismatch site. So to summarize this part, what we did is we developed SAMBA, an assay to measure the effect of mismatches on transcription factor DNA binding in high throughput and very quantitatively. We found that mismatches completely remodel the protein DNA binding landscapes. It's definitely not the case that one could predict mismatch binding from what we already know about binding to Watson-Crick DNA. We found that oftentimes mismatches lead to enhanced transcription factor binding affinity. Since we don't have a lot of time, I didn't go into our data on how mismatches can create super sites. So these are sites bound by transcription factors with affinity higher than what we see for any Watson-Crick site. So affinity levels that you simply cannot achieve in Watson-Crick DNA can be achieved with mismatches. We also show in our paper that we can see increased binding even in non-specific sites where we don't clearly see a match to the binding consensus of the transcription factor. And there also the effects can be quite large, and there's a lot of work to do to understand why this happens. We found that this enhanced TF binding can be explained by mismatches prepaying the penalty to distort the DNA upon binding. And this actually provided insights for us into the role of conformational penalties, which seem to be an important part of protein DNA recognition. And these penalties are something that could not be quantified before, but we can now quantify them by using these specific mismatches that mimic deformations in the bound DNA. We have a lot of other analysis that at least we find very interesting that are described in our paper. But the paper really focuses on the structural aspects and the conformational penalties that we were able to quantify. But as I mentioned, the reason why we went in this whole research direction was because of this hypothesized connection between transcription factor binding and an enrichment of mutations. So does this work help us get any closer to an answer here? It does to some extent. At least it shows us that transcription factors do bind to DNA lesions. In this case, mismatches, and they do so with high affinity. This is an important result because if we saw no binding to mismatches, then there would definitely be no competition to study. But there are still a lot of questions that remain. Given that transcription factors do bind with high affinity to mismatches, is that sufficient to say that they will potentially interfere with recognition of the lesions by repair enzymes? Without actual data, one might actually be concerned about the potential competition here, especially since repair enzymes have very different kinetics from transcription factors, very different ways of searching for these lesions. It is true that at equilibrium, repair enzymes and transcription factors have similar affinities for DNA, but the way they recognize the DNA is very different. So we're very interested in answering this question of whether transcription factors can actually compete with mismatch repair enzymes. And we have preliminary data showing that yes, they can. There's still a lot of work to be done here, and the competition is very complex, as one might expect. So we are working in this direction with Dorothy Erie from UNC and Keith Benninger from NC State. They are just 20 to 30 minutes away from us. And also Alex Drohat from University of Maryland. the preliminary data we have now shows that indeed we do see this competition. It's just a very complex process. We're also trying to understand whether transcription factors bind to mismatches and compete with repair enzymes, but in living cells, because these experiments, initially we do them in vitro, just in cell-free systems. And answering this question is a bit more complicated. We are very interested, we're thinking about some ideas on how to do this and how to use some of the recent technologies to help us in this direction. And we're also very interested in finding collaborators who might be interested in helping in this direction. Even without directly proving that transcription factors bind mismatches and compete with repair enzymes, we can still look in living cells and ask whether this competition potentially leads to increased mutation rates. And we also have an ongoing project in collaboration with Sue Jinx Robertson and David McAlpine here at Duke. They're also in buildings around us. And where we developed a mutagenesis system in yeast. And we're trying to see whether mismatches that are bound by transcription factors really lead to more mutations in the cell. It's not easy to do this given how rare mismatches are, but we're starting to be convinced ourselves that this does happen in the cell. With this, I would like to thank everybody who collaborated and contributed to this work, especially Ariel, who really drove this project. Harshit and Zach in our lab also contributed to the mismatch paper. This was, as I mentioned, a close collaboration with Hashim and his students, Honglu and Atul, and also with Maria Schumacher at Duke and the Poon, Pufol, and Haran labs. We're also grateful to Sue and David for the collaborations on the in vivo work, our collaborations on mismatch repair enzymes, and last but definitely not least, I would like to thank Shira Adar, who was here at who was at UNC when we started this research direction and was really instrumental in the initial discussions. And I also want to thank our funding sources, and thank you all for listening.
Really fantastic talk. We've got about 5 minutes for questions. So if you have questions, please type them into the Q&A box. Our first question is from an anonymous attendee, but I think you basically addressed it at the end. They were asking, could you use an assay like ATAC-seq to try to look at the potential in vivo relevance of potentially, mutations driving, say, transcription factor occupancy? But it sounds like that's exactly what you're not ATAC-seq necessarily, but using other assays to try to figure out how this is working in cells.
Yeah, I think
to add there.
I would add that I think the anonymous attendee is asking about effect on gene expression. And that's something we are struggling with a bit. And it's not a direction that we're following initially, at least, because we do think that because the binding of the transcription factors is affected so much, it could lead to changes in expression. But mismatches or replication errors happen very infrequently. So you might have a rate of 10 to the minus 6, which means you have that particular mismatch in one cell out of 1 million cells. Does that really lead to an effect in gene expression that one can even measure? I'm doubtful. So we think there might be effects in gene expression, but at the level of a population of cells, I don't know if that's physiologically relevant. So it's not really the direction we're focusing on.
Awesome. Okay, and so then the next question is from Klim, who is a QBI fellow and PI here. And Klim asks, are there any TFs that structurally have been seen to bind to the ideal B-form DNA? And if so, do you see that mismatches have no effect on their binding affinity? and then he has a second unrelated question that maybe we could ask after.
So, I mean, we haven't really looked at it that way, but we have looked at the transcription factors that we have in this study definitely have regions in their binding sites where the DNA is nicely B form. It's like an ideal B form. And we definitely see at those positions where the bound DNA is more like B form, mismatches typically decrease binding. So they are detrimental. If the transcription factor really likes the DNA to not be distorted, distorting it can cause problems. And we definitely see, if we look at the positions where mismatches increase binding, those are positions that we see an enrichment in positions that are distorted anyway in the bound form. So where the protein likes distortions, mismatches can increase binding. Where the proteins doesn't like distortions, we mostly see decreased binding. We haven't looked that carefully at where we see no effect, but that's something to look at.
And then I think Klim's next question, which he notes is unrelated. I think my guess is the direction he's thinking about this as a tool. Can you exploit this mechanism to enrich for mismatched DNA? Maybe I can unmute him if I'm
not sure I understand.
I think this mechanism could be exploited to find mismatches for DNA repair. Klim, do you want to unmute yourself and maybe clarify?
Sure. I guess I'm just a little rusty on my protein-DNA interactions, but is this a fundamental way that the DNA repair enzymes find mismatches effectively, where you basically have to have a partially distorted DNA as your binding site, or not?
Yeah, the DNA repair enzymes do take advantage of distorted DNA, and that's one of the main things they recognize. Because if we think of a mutase, it's the main mismatch repair enzyme, it needs to recognize a variety of mismatches. So repair enzymes will care more about the structure of the DNA and how much the DNA is distorted and deformed. So that's already the way repair enzymes work. We didn't expect transcription factors to also take advantage of this.
I guess while I'm unmuted, maybe a follow-up question. Are there any examples where transcription factors actually recruit DNA repair enzymes? Because one could imagine that some sort of residence time of transcription factor, is potentially being read out as a way to find mismatches, or no?
That's a very good question. And that's something we try to keep in mind, because we're always looking at this TF binding to mismatches as something negative that would interfere with repair and make repair more efficient. But we're trying to keep in mind that they could also help with recruitment. Now, I'm not aware of any examples where transcription factors help recruit mutase for mismatch repair. But in other repair pathways, we know there are transcription factors that somehow help with repair. In other projects, we're very much trying to keep this in mind and see if recruitment is possible. And that's something we would see in our assays when we put both proteins together. But we don't have any data for this yet.
Very cool. Thank you.
Thank you.
So I think we're right at the hour. And so maybe with that, please, on your computers, give Reluca another round of applause. Thanks again, Reluca.
Thank you.
Thank you.